Hello and welcome to my lecture on endocrine physiology. This particular lecture video is actually going to be an introduction to some of the principles of endocrine physiology, and then we're going to kind of lead into some of the more specific endocrine organs and glands, and that'll be in the next several videos. So to begin, we have the endocrine system, and the system is it's not as a system as we think of it with some of the other systems, where there's the GI, the GU systems, or the nervous system, muscular system. With the endocrine system, we're really talking about the composite of glands or tissues or cells that secrete hormones and that kind of interact with one another to carry out some sort of biological function. And so you may have a discrete bunch of cells located in one part of the body that communicate with other parts of the body that are much more distal, or maybe communicates with most of the body. So it can be quite widespread or actually very narrow in its function. But one of the keys is that it communicates via the circulatory system, so it releases chemical mediators into the bloodstream. Now, endocrine glands are defined as these organs that are really the traditional sources of hormones that we know of, but the reality is that it's not just glands. There can be actually just cells, like individual cells that can secrete hormones that are not necessarily part of a more composite gland. The hormones, so the chemical mediators that are released into the bloodstream, they're going to stimulate physiological responses in cells of other tissues or organs. And like I said, it could be located fairly close to that particular cell or organ or gland, or it could be more distal. But the action happens in the bloodstream, which means really in order for an endocrine organ to communicate with something that's with another cell or another gland or tissue, that gland or tissue has to have receptors for this particular hormone. And so those receptors are going to be key in the endocrine system in making sure that there is specificity with its communication. So when a gland releases its hormone to the bloodstream, I mean, it's not just targeting one particular tissue. That blood's going to go throughout all the tissues in the body. So the way that the hormone is going to know where to interact, or let me say know where to interact, is based on its affinity for these receptors that are on the surface of these cells, or in some cases receptors that are located within the cells themselves. So when we have this communication between one site and another site, we have this what we call feedback mechanisms, so that the tissues or these organs that are releasing these hormones know when to release more, okay, if it's needed, or to actually decrease its production of a particular hormone. So there has to be some kind of feedback so it knows. And so down here, if you take a look at this cartoon, you have an endocrine cell, all right, and the endocrine cell releases A, so whatever that chemical mediator is, which is labeled as A here. A then goes to its target endocrine cell. Now, again, that could be fairly local or it could be much more distal, but it would be traveling through the bloodstream here. And then that target endocrine cell is going to release its compound, or it's going to react to whatever that particular hormone was. And so if it's releasing something, that may be what's causing the biological effect ultimately. That could be something like vasoconstriction or vasodilation. It could be increasing the heart rate. It could be telling the liver to increase or decrease its production of glucose. So over here we have another example. So this, by the way, this is a feedback mechanism. So what happens is when B is produced ultimately in response to the hormone A, as the levels of B go up, that then feeds back and tells the endocrine cell that released the hormone initially, it tells it to essentially turn that off, or it inhibits the ability of that cell to produce any further. And that will end up turning off this production of A, and therefore it'll stop stimulating the target organ to produce B, and so B's levels will also decline. And that's a classic negative feedback, which is what you'd see in most cases in the body. However, we do also have what we call positive feedback. So what you see here is the endocrine cell releases this hormone or this mediator here, A, into the bloodstream, goes to the target cell. That target cell then has its biological effect. Now, when it releases its mediator here, all right, so it's producing, that carries out this biological effect, B here can do the same things we saw before, but instead of inhibiting its production from the endocrine cell, it actually stimulates that cell to further increase the production of the hormone, which will then ramp up more of the B here, is more of its concentration or more of its biological effect. And so we do have some examples of positive feedback, which we'll discuss in the videos as they come up. Over here, we can also have complex responses. So when we get to this more specific example, this is showing you the hypothalamus communicating with the anterior pituitary, which then communicates with target endocrine cells. Okay, so the hypothalamus, located in the brain, can release its hormone here, okay, which is just labeled as RH, all right, releasing hormone, for example. That'll communicate with the cells in the anterior pituitary. The anterior pituitary will then release its hormone into the bloodstream, which will then communicate with the target endocrine cell. Now, in this case, that could be the thyroid gland, since we'll be talking about the thyroid gland today. So the thyroid, let's say it's the thyroid, then the thyroid will release its thyroid hormone. Now, the thyroid hormone itself that's been released, as that level starts to rise, it can feed back, as you can see, and both inhibit the hypothalamus as well as the anterior pituitary. But if we go one step back, all right, to the hormone that's being released from the anterior pituitary, that could also, those levels can also rise and feed back and inhibit the hypothalamus. So this is more of a sort of a complex regulatory system where we have multiple ways of modulating the response. And we'll talk more to that as we get there. Okay, so when we talk about the endocrine system, again, we're really talking about sort of these discrete organs or cells that are communicating with one another. And there's really what we call the sort of classic endocrine system, which you kind of see depicted in this cartoon here. However, there are many systems that partake in sort of endocrine function, like the GI system, the nervous system, skeletal muscle, and so on. So there's more than just this. It's actually quite an integrated system with many different organs here. But classically and more traditionally, we'll sort of describe these here under the endocrine system lectures. And so that's what I'm going to focus on for this, for these next few videos. So here, kind of working from the top down, in the brain, you can see the sagittal section here, and you see over in this region here, this is the mid-sagittal section, the pineal gland, and the pineal gland makes melatonin, which helps us with our circadian rhythm. Over here, you see the hypothalamus, and the hypothalamus is really kind of a good junction point in our nervous system to communicate with our endocrine system. And so the hypothalamus will communicate with this pituitary gland, which is this little bulbous structure here. You can see connected via a stalk just below the hypothalamus, and we'll talk about that in one of the lectures. Then there's the thyroid gland, which we'll discuss today, and that's located in the anterior aspect of the neck, and actually just anterior to the trachea. And what you're looking at in the cartoon here is actually the posterior aspect of the thyroid gland. And you can see these four little spots right there. All right, these little clusters of cells known as the parathyroid glands, which help to regulate calcium. And then we have, as we work our way down, the thymus, which is involved in immune function. The adrenal glands, located on top of the kidneys, and we'll have a lecture discussing that, but they'll release things like catecholamines, like epinephrine and norepinephrine from the medulla, but they're also involved in things like aldosterone synthesis and cortisol. And then there is the pancreas, and of course, during the GI lecture, I'll discuss some of the pancreatic function. All right, while regulating, for example, insulin, glucagon. And then there in the gonads, you have the ovaries in the female, and you have the testes in the male. And during the reproductive lectures, we'll talk about the hormones that regulate the production of sperm and the ovulation cycle. So hormones have specificity. So as I mentioned in the first couple of slides, when a hormone is released and it goes into the bloodstream, the way it's able to target its tissue is based on its specificity with a receptor. Now that means there may just be specific cells in the body that have those receptors, and so therefore they will be the ones to respond. In some cases, most cells may have a receptor to a particular hormone. For example, like thyroid hormone, interacts with many, many different types of tissue, whereas others that may be more specifically targeted. And so that target becomes specific based on the receptors that are on the surface of those cells. So again, they'll have these specific receptors and or again enzymes for a hormone and can thus respond to it. Now, I should mention that the receptors don't necessarily have to be on the surface of the cell itself. They can actually be located within the cells because, as you guys are going to learn, hormones can either interact on the surface of cells or they can actually bypass receptors and kind of go right through a plasma membrane and enter into the cells, and in which case it can interact with receptors that are located internally. Interestingly, a target cell can contain hundreds to thousands or even 100,000 receptors. And so that's actually going to be another way to modulate the responsiveness to a particular hormone is going to be based on, one, a hormone's concentration in the blood, and also its receptor density. So how many receptors does it actually have? So the number of receptors plays a role in how the cell is going to respond. So over here, you can see this cartoon, you have target cell for hormone A, and you can see it has receptors specifically for A, but it's not going to respond to hormone B because there's no receptor for it. Whereas this particular cell has a receptor for both types. And so you'll see there'll be different tissues that can respond to multiple hormones. And then over here, this one has only the receptors for B and not for A, so it'll respond to those hormones. So this is where the specificity comes in. Now, in terms of the chemical classes of hormones, you have something we call lipid soluble, otherwise known as hydrophobic. So it readily interacts with lipid components. So something that might pass through plasma membranes, for example. So steroid hormones are in that category, and they're all derived from cholesterol. And we'll talk about that pathway in a different lecture. And some of the products of the synthesis from cholesterol is estrogen, testosterone. Again, we'll talk more about those two in the reproductive lectures, but then things like cortisol and aldosterone. Thyroid hormone, interestingly enough, is actually also hydrophobic, so T3 and T4. So these are actually tyrosines, or two tyrosines that are essentially linked together, and they're connected to iodines, and we'll talk about that later. So one of the features of lipid soluble means that most of the time their receptors are going to be internal in the cells, okay? Whereas water soluble, so these will be hydrophilic, so they'll interact more with water. Monoamines, so like amino acid derived hormones, so like catecholamines, also derived from tyrosines similar to the thyroid hormones. So epinephrine, norepinephrine, dopamine, and so on, they're all derived from tyrosine. Then you can also have protein and peptide hormones, so several amino acids linked together, and these will be ones like oxytocin, insulin, antidiuretic hormone. In fact, most of the ones we're going to talk about from the pituitary gland. Then there's the category of the eicosanoid hormones, and so these will be derived essentially from triglycerides, particularly ones that have arachidonic acid, and they can produce prostaglandins and leukotrienes. And their function is quite diverse. So it can actually increase or even inhibit immune responses. It's involved in blood clotting, in vascular tone, and so on. So in terms of hormone synthesis, so the steroid category, the hydrophobics, as I mentioned, derive from cholesterol. So we can produce things like estrogens and testosterone from cholesterol. The monoamines, or the amino acid derived, like tyrosine or tryptophan. So tyrosine, we can produce epinephrine, norepinephrine, dopamine. Combining two tyrosines, we can actually form things like thyroxine, which is one of the thyroid hormones. And you can see some of the iodine that's bound to that, which is there's 4 of them. So this would be called T4. So this is thyroxine. Tryptophan. So that's another amino acid that we can make hormones from. So this one would be melatonin, for example. And then over here we have peptides. So with the peptides, this would be several amino acids, of course, linked together. You can see here we have oxytocin as an example, which is from the pituitary gland. And then over or over here you can see a larger peptide called insulin, which comes from the beta cells in the pancreas, helps us to regulate our blood sugar levels. And here's an example of the hydrophobic pathway. So this pathway here, you can see here's cholesterol, which is the precursor for all the steroids. And you see we can convert that into progesterone, which is the next precursor. And then we'll have several enzymes, which we'll talk about in a different lecture. And those enzymes will convert them into things like testosterone, cortisol, and aldosterone. Okay, so this is an example of hydrophilic. So I talked about the monoamines, but they're not depicted here. And this will be one of the peptides. And you can see this is the insulin example. And so interestingly enough, with insulin, it's produced in the beta cells in the pancreas. And what you see here in this peptide in the yellow is the insulin molecule. And then it's connected via a connecting peptide. And actually, this is how it's produced inside the cell. And then in the Golgi, it gets disconnected, and so it removes the connecting portion of it. However, when the cells are stimulated to release insulin, insulin is released in equal amounts to connecting peptide. Now, the reason this is interesting for us clinically is, let's say somebody has diabetes and they're taking insulin, okay? And we want to know how much function the beta cells still have. Well, we would see how much connecting peptide there is, because the concentration of connecting peptide should equal the amount of insulin that they're producing, because they're produced in equal molar amounts. So it helps us to figure out endogenously how much insulin the beta cells are actually producing. And so we can take a look at that connecting peptide. If they're taking insulin, that's exogenous. That insulin doesn't come with a C-peptide, or doesn't come with a connecting peptide, excuse me. So we can't measure that. So the C-peptide, or the connecting peptide, actually acts as an endogenous marker of insulin production, which can be very helpful for us in terms of keeping an eye on the beta cell's function. Okay, so when a hormone is released, if it's hydrophilic, that hormone, so over here in A, hydrophilic, when it's released, it can dissolve readily in the blood plasma and be transported in what we call a free or unbound state. The hydrophobic hormones, on the other hand, when they're released into the blood plasma, they're going to readily, or typically readily, bind to proteins present in the plasma. So, and the proteins will help to transport them and keep them in a dissolved state. So not only does it help them to dissolve, but there's also something called a dynamic equilibrium. So what this means is that when a hormone or hydrophobic hormone is released into the plasma, the majority of that hormone is going to bind to the protein, like 99% or more, which means not all of it will bind. So there'll be what we call the unbound or free portion of that hormone. So that's usually like 1% or less for many hormones. And so what you have is an equilibrium between what's unbound and what's bound. And so if the unbound or the free hormone is getting used up by the cells, okay, or interacting with the cells, then more hormone will actually unbind from the proteins in order to keep that equilibrium. Okay. Same thing goes if you have an influx of hormone into the plasma, then more of that will then 99% of that will then bind to the protein and keep that unbound proportion fairly even or fairly consistent. So here in this cartoon, what you can see is we have the hydrophilic, and so this is an example of a peptide, and the peptide can actually bind to a surface receptor, again, because it can't pass through the plasma membrane on its own, and that can actually cause a cascade of events inside the cell, because what will happen is if it binds to the receptor, this can initiate internally changes within the cell that create what we call a cascade of second messengers. So in this example, like it can produce or it can activate an enzyme located in the membrane, which can then convert ATP into cyclic AMP. Cyclic AMP then can move inside the cytosol of the cell and activate enzymes. And those enzymes, depending on what those enzymes are, will have various metabolic effects. Okay, so second messengers like cyclic AMP or IP3 are very common. Then we have hydrophobic. So hydrophobic would be, again, here's our steroid molecule, or called a cholesterol backbone, if you will. And so that can actually pass readily through the membrane and interact with receptors actually located within the nucleus. So not only does it pass through the cell membrane, but also the nuclear membrane, goes inside the nucleus and can interact with specific receptors there, which can modulate gene transcription and ultimately translation. So if we get to messenger RNA and protein synthesis, it can actually produce proteins. This particular pathway, however, tends to be slower than the hydrophilic pathway. The reason being is it takes time to actually transcribe and then translate and produce enough proteins to have a biological effect. And so therefore it can usually take a little bit of time. In terms of our cyclic AMP second messenger system, this would be how hydrophilic hormones interact with surface receptors. So over here we have our hormone, and here's a box. This box is just indicating all the different types of hormones that would fall in this category of hydrophilic that might interact with specifically a cyclic AMP second messenger system. We'll talk about all these different hormones at some point or another, but just to give you an idea of what some of them are. Adrenocorticotropic hormone, ACTH, or follicle stimulating hormone, and thyroid stimulating hormone down here, and calcitonin and glucagon. Catecholamines like epinephrine and norepinephrine and dopamine, they would all interact with surface receptors. And when the hormone binds to its specific receptor, like for instance if it's epinephrine, it could bind to adrenergic receptors like alpha or beta receptors. When it interacts with it, again, you might have what we call this G protein linked receptor. So it interacts with this and interacts with its receptor. There's a conformational change in the G protein. It switches a GTP with the GTP for GTP, and that dissociates. Interacts with a nearby enzyme called adenylate cyclase, which converts ATP into cyclic AMP. Cyclic AMP then diffuses into the cytosol, where it can actually turn inactive protein kinases into active protein kinases. And a kinase is something that phosphorylates something else. And so that can actually take enzymes that require phosphorylation in order to be active and turn them on, or it can actually phosphorylate an enzyme and turn it off. It just depends. And then the enzyme can then act on its particular substrate, and what you have is essentially kind of a cascading effect, and this can ultimately produce a metabolic effect within the cell. So this is also a second messenger system, but this one's looking at the IP3 DAG second messenger system. And so hormones that might interact with receptors that initiate this second messenger system are ones like antidiuretic hormone, for example, or thyrotropin releasing hormone. And so when that hormone binds the receptor, again, it's G protein coupled, and we have that subunit, but this subunit now is interacting with a phospholipase enzyme, and the phospholipase actually cleaves one of our phospholipids that's actually in the membrane there and converts it into a diacylglycerol. And then there's an IP3, or an acetyl triphosphate, and the IP3 actually diffuses inside the cell. And one of IP3's main functions is actually to interact with the endoplasmic reticulum on these receptors that release calcium. So calcium is usually stored inside these endoplasmic reticulum. That gets released into the cytosol and can interact with things like calmodulin, and that can interact with other kinases. We've discussed that in terms of its action on myosin light chain kinase, for example, where calcium plus calmodulin plus the myosin light chain kinase can actually activate smooth muscle contraction. Then you have diacylglycerol, which stays membrane bound here, but can convert protein kinases and activate those protein kinases to interact with and again phosphorylate particular enzymes and increase their activity or decrease their activity. So again, as I kind of mentioned, smooth muscle contraction, protein synthesis, secretion, mitosis can all be regulated by these second messenger systems, and also then regulated by these hormones. So interesting when it comes to hormones, they can have one particular hormone, for example, can have a whole diverse or array of actions on various tissues, or even in the same cell. So as an example of that, if we took a look at, say, epinephrine. Now, epinephrine can interact with adrenergic receptors as well as beta adrenergic receptors, so alpha and betas. Depending on the tissue that it's located on, the cell it's located on, it can carry out different functions. So, for example, epinephrine acting on alpha receptors in smooth muscle of blood vessels causes it to vasoconstrict, whereas epinephrine acting on beta adrenergic receptors of smooth muscles can cause them to actually vasodilate or relax. Meanwhile, if epinephrine interacts with beta adrenergic receptors on the heart, it can increase heart rate and contractility. So, but these will be like different subtypes of these receptors. But again, this epinephrine having multiple effects on different tissues. In this case, we could even have it, interacting with an alpha and a beta. And in the case of this alpha receptor, it's connected to an IP3 messenger system. This one over here, the beta, is connected to a cyclic AMP. And you can even have it funneling into a very similar effect, which in this case is to break down glycogen into glucose 1-phosphate. So this might be an interaction that you'd see with epinephrine on, say, a liver cell or a hepatocyte, because the liver cells, or the liver, I should say, is one of its functions is to regulate glucose levels, and epinephrine is known to actually interact with hepatocytes and increase glucose release from hepatocytes into the blood, so we can increase our blood glucose levels that way. Now, there's also a term here which I've mentioned in the title called pleiotropic effect. And so a pleiotropic effect is essentially a single hormone having multiple effects on the same tissue. So a good example of that would be insulin, for example. So insulin on skeletal muscle. If insulin interacts on skeletal muscle, it interacts with a specific receptor. And what can happen, though, is to the skeletal muscle, it could actually increase glucose uptake, which would be one function. It can also increase glycogen synthesis, so it can store the glucose. It can also prevent glycogen breakdown, so it inhibits that. It can increase amino acid uptake and protein synthesis and inhibit protein breakdown. So these are all functions from insulin. So it's having multiple effects. And so we would call that this pleiotropic effect. So insulin is a good example of that. Now, the hormones also are very, very low concentrations. In other words, they're very, very potent, which I have over here kind of highlighted for you. These are very potent chemicals, meaning it doesn't take a very large amount to get a very large biological response. Just to give you an idea, we're talking concentrations on average somewhere in the nanomolar amounts to even like picomolar amounts. We're talking about like 10 to the negative 9th amounts. So these are very small concentrations, where you could have a single hormone surrounded by 50 billion water molecules. So it stands to reason that one of its mechanisms of action would be that when it binds to a receptor, is that the response inside the cell is to amplify that response. So you can have a single hormone. So if I have a small stimulus for, let's say, a small amount of hormone, that hormone is going to then amplify the internal effect on the cell. So what happens is a lot of cyclic AMP is produced, and you get a lot of protein kinases that are activated, much more relative to the amount of hormone that actually bound. And then those kinases can activate downstream enzymes, and it can activate a whole lot more enzymes. Okay, and then all those enzymes acting on their substrates can produce a lot more metabolic product. So from a small amount, you get a large response, and we call this process amplification. And it's a necessary mechanism for hormones, and the hormones, again, they come in very small concentrations, so this is a very potent effect. So there's something else called receptor regulation. So when it comes to hormones, one factor is the concentration, which I have down here in bold. So the cellular response is governed by hormone concentration, so that's one factor. All right, and then you have the receptor's affinity. So, how avidly it binds this particular hormone. And then, of course, the amount of the receptors that are actually present. So these are really three factors. The amount of hormone, the amount of receptors, or the number of receptors, and the affinity of those receptors for that hormone all dictates how a cell really responds to the hormone. So we'll keep that in mind. So now, if you take a look at this cartoon here, we have something called upregulation, and down here is downregulation. So upregulation, we take this cell as an example. We can have a fixed, let's say, concentration of hormone, and it has a certain number of cells, and those, oh, excuse me, certain number of receptors, and those receptors have a certain affinity for this hormone, let's say. So those are the three variables we just talked about. So if it's stimulating these particular receptors, the cell's response might actually be, in some cases, to increase the number of receptors. This is going to ultimately change the overall cell response over time, because even if you have the same fixed number or fixed concentration of hormone and the same affinity, now that I have an increased number of receptors, I can actually increase the responsiveness of that cell. And we refer to that as upregulation, which will occur in some cells, but not all. In other cases, you may have, a certain hormone concentration, and here's our receptors, all right? And in response to that certain concentration, over time, what that cell will do is actually to remove those receptors from its surface so that there's fewer of them. And this is referred to as downregulation. And so in this case, the cell becomes less responsive over time. And you can actually see this effect not just with hormones in our body, but also medications that patients take. Sometimes at a certain concentration, the receptors actually downregulate, and you may have to change the dosages of certain medications to increase the responsiveness. In addition, hormones can influence another hormone's receptor. So there's going to be some other interplay. So for example, a thyroid hormone can increase adrenergic receptors on cardiac myocytes. So let's say there's an increase in thyroid hormone stimulation of the heart. The heart will increase the number of, let's say, beta receptors. More beta receptors means it's going to become more responsive or more sensitive to epinephrine, so more likely to increase heart rate and so on. Estrogen, another example, it can increase progesterone receptors in the uterus, so it makes the uterus more sensitive to the effects of progesterone. It can also decrease parathyroid receptors on the bone, and when we talk about calcium regulation, we'll talk about sort of the significance of that. Some other factors to keep in mind: exercise can increase insulin receptors on the surface. So it can increase sensitivity to insulin, which is why it's actually an important thing to tell patients, especially pre-diabetics, to increase their exercise, because if you can increase the number of insulin receptors, if they are low on insulin, this might actually increase their ability to, or increase their sensitivity to the insulin. Obesity has the opposite effect. So obesity actually can reduce insulin receptors and make them less sensitive to insulin over time. So it's the same basic principle, where the cell's response is governed again by hormone concentration, receptor affinity, and number of receptors. So the examples I've kind of been giving you is by changing cell receptors, but it can also change affinity. So in some cases, it can modify a receptor's affinity by tagging it with something like a phosphate group or something else like that and reduce its affinity. So there will be other mechanisms that can come into play here. But overall, these are the three variables in which we can vary cell response. So most cells are sensitive to more than one hormone, and they'll exhibit what we call interactive effects. So one of the effects is synergistic, and so that's really where they're acting together for a greater effect. So an example of that would be follicle stimulating hormone and testosterone acting together for sperm production. The example I gave on the previous slide, which is thyroid hormone stimulating the heart and acting synergistically with, say, epinephrine to increase heart rate and contractility and so on. A permissive effect would be one hormone enhances the target organ's response to a second later hormone. Estrogen prepares the uterus for actions of the progesterone during pregnancy. So estrogen actually increases the number of progesterone receptors on the uterus, and so when estrogen levels start to decline and the progesterone kind of takes over, its effects later on are more pronounced. Another example of that would actually be in the case of cortisol. So cortisol can actually increase smooth muscles sensitivity to angiotensin 2. So therefore, cortisol levels can can have the effect, if there's an increase in cortisol, have the effect of causing hypertension, because angiotensin 2, if it interacts with the smooth muscle cells, causes constriction, which can increase blood pressure. Antagonistic effects would again be opposing effects, and so we see quite a bit of that. So that would be like estrogen from the placenta opposes prolactin, so prolactin is not produced. Insulin and glucagon, for example, are sort of opposing. Insulin lowers glucose levels, and glucagon helps raise it. Parathyroid raises calcium levels, whereas calcitonin lowers it. So those would be antagonistic examples. So to kind of wrap up sort of general information about the endocrine system, there's something called hormone clearance. And so this is, really in the body, it's about the rate of production of hormone, how much is being produced and released into the blood. And that has to be balanced with the breakdown and removal of that hormone. And as long as we're in an equilibrium, or essentially what we call a steady state, where we're producing and we're breaking down essentially in equal amounts or at an equal rate. And so we reach what we call a steady state between the production and the breakdown. So most hormones are taken up and degraded by the liver and the kidney. And so those will be our two primary organs. Primarily it's excreted in the urine, but sometimes also in the bile, which will enter into the digestive tract and then be excreted as feces. Hormones will typically either be metabolized into different molecules. So you'll take whatever hormone it is and break it down into totally different molecules that get excreted, let's say, in the urine. And so those would be metabolites of that particular hormone. In some cases, instead of metabolizing the hormone into different component parts, you might just modify it by covalently linking it to something else, to another molecule, to inactivate the hormone. So a classic example of that is glucuronic acid. So the liver, for example, can combine glucuronic acid to, say, a hydrophobic hormone. And glucuronic acid actually makes it more charged, so it's more soluble. If it's more soluble, if it stays in the plasma, we can filter that more readily in the kidneys there, and it can be excreted that way, or in the bile. And then there's something called the MCR, or the metabolic clearance rate. This is analogous to the renal clearance, which I have on a different video. Ultimately, it's the same concept, which is the volume that's been cleared of a hormone over time. Okay, so in terms of what I want to really just discuss with that is half-life and clearance are inversely related. So what happens is the kidneys can maybe clear a certain amount of blood of a hormone over a given amount of time, and it can ultimately be excreted into the urine. And if the clearance of a particular hormone is very high, meaning it's being removed pretty readily, that means the half-life of a particular hormone is very short. So that's how they're inversely related. So again, if the clearance is very low, meaning I'm not removing a whole lot of that drug over time, I'm not removing it from the plasma, that means that its half-life is longer. So there'll be certain properties that it might have that might make it more or less likely to clear. So for example, if a hormone binds to proteins, like a lot of the hydrophobics. The hydrophobic hormones, they have longer half-lives generally because when they bind to proteins, they don't filter as readily through the kidneys, for example. So that generally tends to lengthen their half-life. So their clearance is low and their half-life is greater. Okay, so we're going to start actually talking specifically now about the thyroid gland. So a couple of interesting facts. The thyroid gland is the largest purely endocrine gland. It's composed of these two lobes over here. So it's got these two lobes, which is connected by this isthmus right here. And this is on the anterior aspect of the trachea, just below the larynx. Here's our voice box right here. So just below that, you can actually feel your Adam's apple, which is this thyroid cartilage here, right around there. So just below that is your isthmus. Okay, so that can be palpated, and that's something you'll palpate, and you'll learn to do that in the physical diagnosis class. All right. Now, the thyroid gland is composed of something called follicular cells, because there are what we call a lot of follicles in the thyroid. And so these follicles are made up of follicular cells, and these follicular cells secrete thyroxine, which is T4, and triiodothyronine, which is T3. The number simply just indicates the number of iodine molecules that are bound to it. So T4 has 4 and T3 has 3. There are also parafollicular cells located in the thyroid gland. Parafollicular cells are also called C cells or clear cells. They secrete calcitonin. I will not really be discussing this in this lecture. I'll be talking about the parafollicular cells in the calcium lecture. Okay, so this will be more regulating calcium by lowering calcium levels. It'll do that by stimulating osteoblasts on the bone, so it'll bring the calcium from the plasma and deposit it onto the bone. So more on that in the calcium lecture. So the thyroid hormones, all right, again secreted as T4 and T3. Generally, the follicular cells will secrete a higher proportion of T4 than T3. In fact, the ratio is about 20 to 1. T4's half-life is about 6 or 7 days, which is longer than T3's half-life, which is about 1 day. The thyroid hormone is transported in the blood bound to proteins, again because it's hydrophobic. So it's bound to thyroxine binding globulin, which is called TBG. And actually 80% of thyroid hormone is bound to thyroxine binding globulin. There are also the sort of the remaining 19% or so is bound to transthyretin as well as albumin. And then there'll be about 1% or less will be unbound. So that's that dynamic equilibrium I was talking about. And so the unbound fraction here is actually the part that's biologically active. That's the one that will interact with the receptors inside the cell. So to interact with the cell, again, since it's hydrophobic, it will generally pass through facilitated transporters. It'll interact with intracellular receptors. So in this case, when you're looking at the cartoon here, here's our T4, which is predominantly how it's carried in the blood. The T4 enters through facilitated transport inside the cell, where there's actually an enzyme that'll remove one of the iodines and convert it into T3. So here's the kicker, is that T4, although that's primarily what's secreted by the thyroid follicular cells, the tissues that respond to the thyroid hormone convert T4 into T3 because T3 is the biologically active form. So T4 is converted to T3 inside the cell. T3 can then actually move through the nuclear membrane and interact with receptors inside the nucleus. So it binds to its receptor inside the nucleus, which can then again modify gene transcription and translation, and that will increase the amount of messenger RNA that's produced in protein. And for the most part, this will make the actions of thyroid hormone a little bit slower. It takes on the order of usually days for an effect to be seen, in most cases, but not all. Again, this is just anatomy showing you the thyroid gland. If we take a look at the cadaver, we cut away the anterior aspect of the neck, like the platysma and so on. You can see here, highlighted in purple, is this the lobes on either side, the isthmus, and sometimes they'll have this pyramidal structure. So some people will have this what we call sort of pyramidal lobe here. If we were to actually dissect the thyroid gland and take a section of a histological section of it, you would see these follicles. So you can see one follicle right here. Here's another follicle over here. All right, so you can see about four follicles and maybe some little ones over on the side there. And these are lined by follicular cells. Okay. Now the follicular cells surround the outside or the circumference here of the follicle, and the inside here is made up of something called colloid, which is a sort of proteinaceous gel-like substance. And so this jelly-like substance is a thyroglobulin. So that's the primary protein located here. This is extracellular. This is not inside the cell. So the cells actually line the follicle, and the thyroglobulin gets released by the follicle cells into this composite colloid mixture here. Okay, and then the blood vessels are actually going around the follicles here. All right, they surround the follicles. And then over here, this is what we call the C cells, or the parafollicular cells. Parafollicular because they're around the follicles. And again, those will be regulating calcium. So if we were to actually take a look at this cartoon here, you can see one of the blood vessels. This is a follicle, so a single follicle, and is lined by the follicular cells. And then the center portion here would be that colloid that we were looking at, that pinkish looking stuff on the histological slide. And so there, if we blow up this little portion here, you can see here are the follicular cells. Here's the blood vessel. This is the colloid over here, that suspension there. And so these follicular cells, this would be the basal surface, the surface that communicates with the bloodstream, and then the apical surface, which is the part that communicates with the colloid mixture. And so you can see just from this right here, we get the iodine. The iodine is required for thyroid hormone synthesis. So iodine has to go through the basal side of the follicular cell, be transported through the apical side, where it's going to then interact with proteins that have tyrosine residues, and the iodine will bind to those tyrosines. Okay, and then it can be taken up by the cells. Over here, it's taken up by the cell, and it can be broken down and cleaved off those tyrosine residues to make your T3 and T4, which can then be released into the bloodstream when it's needed. And then over here, this is just showing you the production of the what we call the thyroglobulin, which is the protein, the primary protein found in the colloid that contains lots of tyrosines. So if we look over here, this little beaded structure here, these are different amino acids, okay, of the thyroglobulin molecule. That's that primary protein in the colloid mixture, and it has a lot of tyrosine residues or tyrosine amino acids. And you can see one tyrosine over here, another tyrosine over here, and there's an iodine bound to one of these tyrosines, but there's two iodines bound to this tyrosine. So you'll notice the name MIT and DIT over here. So this is monoiodotyrosine, meaning that this tyrosine only has one iodine bound to it. And this one is diiodotyrosine, which just means that there's two tyrosines bound to that. So what happens is you have a chemical reaction that occurs inside the colloid there. So again, just to remind you, the iodines, so this, I should say, the thyroglobulin that's been produced by the cell is secreted into this colloid. And the thyroglobulin there is then modified when the iodine enters into the colloid. There's an enzyme that will actually add those iodines. And then another enzymatic reaction that's going to connect these two tyrosines together. So if it connects a MIT with a DIT, you end up with a T3 because there's three iodines. On the other hand, it might connect two diodotyrosines or two DITs, and you get four iodines, and that's going to be T4. And so we primarily produce T4 in comparison to T3 in like a 20-to-1 ratio, as I mentioned. And then T3 is the primary biologically active thyroid hormone. So here, this cartoon is really showing you the synthesis. So over here, this would be the basal side. This is where the blood is, outside of the follicle. And then this is the inside of the follicle with that colloid mixture. So here in the blood, we get the iodine. And sodium is co-transported with the iodine. So iodine gets inside the cell, and it quickly diffuses across the cell to the apical side, where there's a receptor called pendrin, which transports the iodine into the follicle, into the colloid mixture there. At the same time, this cell has been producing thyroglobulin. So it produces this protein. You can see all the different tyrosine residues connected to it, and that gets secreted into that colloid mixture over here. And so what we have now is the iodine and the thyroglobulin in this colloid space here, and through an enzymatic reaction, you're going to get iodination of those tyrosine residues, adding either one or two iodines to it through what we call a peroxidase enzyme. And so this process of adding iodines to tyrosine is known as organification. Now, once you've added these iodines to these tyrosine residues, then there's another enzymatic step where the tyrosine residues are connected to each other. And so again, that's that MITs and the DITs. So the monoiodine tyrosine over here connected to a diiodotyrosine, so that creates a T3. And over here, we've produced a T4. It still stays bound to the overall protein structure. And in this state, it can actually stay stored in this colloid mixture for long periods of time. And then if the thyroid is stimulated to release its thyroid hormone, what's going to happen is you're going to get endocytosis of this molecule. Really, it's like a pinocytosis. It takes that into the cell, engulfs it. That's going to actually fuse with a lysosome, which is going to actually have the effect of proteolysis, where it's going to break down and break off our tyrosine residues from the protein compound there. And we'll just take the breakdown products of that protein and recycle it, and we'll release the products of T4 and T3 into the bloodstream via exocytosis. And that'll happen again in response to a stimulation. So this pattern is very important to recognize in terms of feedback mechanisms for the thyroid hormone. So what we have is TSH, otherwise known as thyroid stimulating hormone. So thyroid stimulating hormone, it's in the name. So what happens is the sort of chain of events is this. This is the hypothalamus in the brain. And the hypothalamus is going to release TRH, or thyrotropin releasing hormone. So when it releases this hormone, it interacts with the pituitary gland. So this is actually called the anterior pituitary. And so TRH stimulates the anterior pituitary to release TSH, which is thyroid stimulating hormone, which I have written over here, TSH. Thyroid stimulating hormone is, as its name implies, it stimulates the thyroid gland. It stimulates the follicular cells. It's going to bind to receptors on the follicle cells. Now, when it does that, the thyroid gland, which is going to release T3 and T4. T3 and T4 will then diffuse into the bloodstream, bind to its particular proteins, and then interact with cells on their receptors. In addition to interacting with cells throughout the body and creating its biological effect, it's also going to, the blood's also going to be returning to the brain, in which case this T3, as the levels go up, is going to inhibit or prevent the release of TRH and TSH from the anterior pituitary and from the hypothalamus here. So this is a negative feedback. As the thyroid hormone levels rise, you get a reduction in the stimulation of TSH to the thyroid. So that's our negative feedback. And this is a very important one to recognize for us, especially clinically, this pattern. So a couple other things about TSH, besides the fact that it releases or causes the release of T3, T4, it also has a growth or growth stimulating effect on the thyroid glands, on the follicular cells. So it can cause hypertrophy and hyperplasia. Again, hypertrophy means an increase in the size of the cell, and hyperplasia means an increase in the number of the cells. So it does have a stimulatory effect, and if TSH levels were to rise, okay, and it was done so chronically, it can cause an enlargement of the thyroid gland itself because it increases protein synthesis and growth of those cells, and it can cause something we call a goiter, which I'll talk about a little bit more later. Synthesis of thyroid globulin. So TSH stimulates growth. It also stimulates the synthesis of the production of thyroid globulin by increasing, the uptake of iodine into the cell, the movement of iodine into the colloid mixture, the production of thyroid globulin, as well as enzymatic activity to combine iodines to the tyrosines and so on. It also increases synthesis and secretion of T4 and T3, so it's going to increase pinocytosis and removal of those thyroid globulin molecules, breakdown and release of the T3 and the T4. Okay, so in terms of sort of general function of the thyroid, thyroid targets most tissues of the body. Okay, so for example, like it does target things like the liver, the kidneys, the heart, skeletal muscle, but has less effect on, say, the adult brain, less effect on, say, the skin or on the testes, for example. These ones would be maybe less responsive. Its onset of action is typically slow, and that's again based on its mechanism of action, which again is to interact with receptors inside the nucleus and cause gene transcription, translation, and proteins synthesis, which takes time. It does influence our metabolic rate, our BMR, which you guys may probably have heard of, but the BMR really is a measure of our oxygen consumption. All right, how much, how much we consume. So if you have a very high BMR, it means you consume a lot of oxygen. But it's not again, it's not a perfect system in terms of its oxygen consumption. So the system tends to produce heat as a byproduct of metabolism. So when you're consuming oxygen, we're also producing a lot of heat. So somebody who has high metabolism generally produces more heat. They give off more heat. And we call this a calorogenic effect, or thermogenesis. So for example, the basal amount of thyroid hormone that we have in our blood dictates our BMR at rest, our basal level, our basal BMR, our basal metabolic rate. If the level of thyroid hormone were to increase, our BMR would go up along with it. It would increase that, so it would increase thermogenesis, which means it would also increase the amount of oxygen consumed and the amount of heat produced. And the opposite is true. So again, that would also promote weight loss or weight gain. So if we have more thyroid hormone around, that would promote an increase in our BMR, and that would promote weight loss. Whereas a reduction in thyroid hormone would cause weight gain by the same logic. One of the mechanisms with which it does this is to actually increase the sodium activity of the sodium-potassium pumps, which helps increase gradients and helps with cellular activity. It also increases the production of some of the mitochondrial proteins that help along the electron transport chain and oxidative phosphorylation properties. In fact, over here on the curve, if you look at the basal metabolic rate, you can see this is the x-axis over here is over days. So for example, if you were to inject thyroxine, you'll notice that it does go up. The basal metabolic rate will go up, but it takes days to see a peak effect. And again, that's due to its slow mechanism of action. In addition to increasing or decreasing our BMR, it also increases or decreases the metabolism of carbohydrates, proteins, and fats along with that, right? And that means, in terms of metabolism, that means both catabolic and anabolic reactions. So it can increase protein synthesis and also increases proteolysis along with it. So it'll increase all of those, and it's directly related. So if thyroid hormone levels go up, metabolism of carbohydrates, proteins, and fats goes up along with it. If we're to look at a specific organ like the heart in the cardiac, right, it can increase heart rate and contractility. And we talked about that in the earlier slides, where it has sort of a synergistic effect because it can increase the number of beta-adrenergic receptors on the heart, which then make it more sensitive to epinephrine, for example. If we take a look at this, it's the daily thyroid hormone secretion and BMR. So this is a percent change in metabolic rate, our basal metabolic rate, or our BMR. And this is how it responds to the concentration or to the amount of thyroid hormone that's being produced. So if this is, let's say, our sort of steady state, if you will, at 0 here, okay, this would be our steady state basal metabolic rate, and this would be sort of our normal thyroid hormone production rate over here, which is close to about 100 micrograms a day. And you'll notice the curve is not exactly linear, because if the thyroid hormone production rate actually decreases, let's say it goes down 10%, let's say the basal metabolic rate goes down 10%, that's a decrease of about, let's say, 100 micrograms to about 50 micrograms or so. If we increase the thyroid hormone production rate by 50 micrograms, let's say about 150 micrograms, that'll increase our basal metabolic rate about 10%. So there's a range, whether we drop 50 micrograms or increase 50 micrograms. Within that range, we're either increasing or decreasing our basal metabolic rate by about 10%. But what happens, though, if we drop further than 50 micrograms? If we go below that, say 25 micrograms, if we go to 25 micrograms, you'll see that we drop to about 30% of our basal metabolic rate. So it drops very quickly. Whereas if I go up to, say, 175 micrograms, or even 200 micrograms, at 200 micrograms, I'm not even at 20% yet. So it's much more responsive to drops in terms of percentage, in terms of its percent effect on basal metabolic rate. It's more sensitive to a drop in the hormone concentration or hormone production rate than it is to an increase. Okay, so another function of the thyroid is for growth and development. So the fetus requires thyroid hormone in order for appropriate development of the central nervous system. It also increases production of growth hormone and IGF-1, insulin-like growth factor 1, which both of these are required for normal growth and development, especially during adolescence. And so thyroid hormone, actually, if the thyroid hormone levels are low, growth hormone levels are also low, because thyroid hormone influences an increase in growth, or induces an increase in growth hormone production from the anterior pituitary cells. So those two are linked via that sort of relationship. So if you have a low thyroid hormone levels associated with low growth hormone levels, you can also have mental retardation. So if it's occurring during fetal development, there's low thyroid hormone, the central nervous system doesn't develop normally. And so what can end up happening is you can end up with mental retardation, stunted growth, usually very small, and some of the features you can see over here, something called cretinism, when there's, when the fetus has been hypothyroid, they don't get normal development of the central nervous system, and you can see some of these other features too, like an enlarged tongue, and then small stature, and so on. So in the adult, however, the ultimate levels of thyroid hormone have been associated with an increase or decreased neuronal activity. So high levels of thyroid hormone have been correlated with anxiety, and low levels with depression. Albeit that, it's not entirely clear how thyroid hormone interacts with the adult brain. Thyroid disorders, there's hypothyroidism and hyperthyroidism. So in hypothyroidism, you're not producing enough thyroid hormone. And in infants, as I mentioned in the previous slide, that can develop into cretinism. Again, because the central nervous system requires thyroid hormone for normal development. So that would be dwarfism and mental retardation. But important to understand that actually, if it's caught early on and treated by giving thyroid hormone in the first few months of life, you can actually prevent things like mental retardation in the newborn. There can also be what we call a congenital lack of thyroid, can cause sort of this hypothyroidism or an iodine deficiency, maybe in some of the third world countries that don't have a diet sufficient in iodine, and that can lead to this hypothyroid state in the infants. In adults, there's it's associated with depression. Like I said, its effects on the nervous system, we can kind of depress some of its activity. It can reduce reflexes. The person can feel kind of weak and sluggish. The muscles just overall feel fairly weak because it's not metabolizing at the normal rate, and they can also feel hypersomnolence and very sleepy. Typically, there's weight gain, so they're usually storing more readily. They have coarse, what we call coarse or brittle hair, and usually cold intolerance. And again, the cold intolerance is associated with the low metabolic rate, because metabolism produces heat, and if they're not metabolizing, they're not producing usually a lot of heat. Hyperthyroidism, particularly, when we're talking about the adult, you're seeing tachycardia, all right, elevated heart rates, weight loss, and sweating. Again, sweating because they're producing a lot of heat from all that metabolism. Weight loss is not always seen in these patients because oftentimes with an increase in metabolism, it also increases their hunger, so they may eat more. Anxiety, because it stimulates the nervous system to be a little more active. Heat intolerance, again, because they're producing a lot of heat from all that metabolizing. Now, Graves' disease over here, which you can see in the picture of this lady here with this condition called exophthalmos, with these bulging eyeballs. And you can see she has a goiter in her neck. So in Graves' disease, this is the most common autoimmune disease that causes hyperthyroidism. So it's autoimmune. What happens is you have the production of a thyroid-stimulating immunoglobulin. And what it does is it actually acts like thyroid stimulating hormone, or TSH. and it stimulates the same receptor. However, it doesn't have the same feedback mechanism. So whereas TSH comes from the pituitary, stimulates the thyroid releases thyroid hormone then should feed back on the pituitary and inhibit its release of TSH. So that's the normal function. The TSI, on the other hand, doesn't have a negative feedback. So when it stimulates the thyroid gland, it causes an increase in thyroid hormone production, and the elevated thyroid hormone production, although it's negatively feeding back on the pituitary, that's not where the TSI is coming from, and so the TSI continues to stimulate it. And since TSI acts like TSH, you can get a goiter. There's growth of the thyroid gland and an increase in thyroid hormone production, and produces a lot of the symptoms I just talked about here. Okay, so when it comes to some of the features of hyper and hypothyroidism, they can present with a goiter and something called myxedema. Now, myxedema is really a change, a dermatological change, something that happens to the skin during hypo or hyperthyroidism. But more commonly, myxedema is usually sort of synonymous with severe hypothyroidism, even though it can really be found in both. And what it really is, it's swelling. And one of the more common swellings you see in the skin is a swelling in the pretibial area, right in the shin. And so they'll call it pretibial myxedema. And so, but it can happen in either hyper or hypothyroidism. And what happens is really you get a deposition of collagen and chondroitin sulfate that's deposit in the extracellular space, and it can hold on to more water. And so it can become sort of a non-pitting type of edema. And usually treated with, oral thyroid hormone if the person's hypothyroid, or or removal of the thyroid gland if it's causing hyperthyroidism. And it's usually associated with high TSH levels, because TSH tends to stimulate this production of myxedema. In, say, Graves' disease, TSI could also do that. So in Graves' disease, you could also see myxedema. Now, a goiter, which you can see over here, in this lady here. So this would be a very severe example where that could have a mass effect, where it could actually be compressing structures in the neck. But this is any pathological enlargement of the thyroid gland, and it's also associated with an elevated TSH level, because the TSH stimulates the thyroid to grow. So you can have what we call an endemic goiter. So this disease occurs in a geographic locality. So really what's happening is you have, let's say, a dietary deficiency of iodine. If you don't get enough iodine in your diet, what happens is it's not enough iodine to supply the production of T3 and T4 that may be needed for your body. And so what happens is, if you don't produce the T3 and T4, that means your levels are low. TSH levels will start to rise. So TSH, because it's not getting any negative feedback, so TSH stimulates more and more of the thyroid gland, and it starts to produce a goiter. And that's due to, again, in this case would be iodine deficiency. And actually, in more developed countries, they usually combine iodine with the salt. So it'll be like iodinized salt, so you can get your daily intake that way. So that can also cause, so again, that's a form of, say, hypothyroidism causing a goiter. Hyperthyroidism can also cause a goiter, because you saw that in the case on the previous slide with Graves' disease, where you had TSI, which is acting like TSH and causing overstimulation. Another example might be if, let's say, there was a tumor in the pituitary gland that's overproducing thyroid stimulating hormone, or TSH, then it could automatically just produce lots of TSH, and usually tumors don't respond to the normal feedback mechanisms, so it would keep the TSH levels elevated, and it might cause more overstimulation of the thyroid gland, causing a goiter as well as increase in thyroid hormone. So let's see if we can quickly just put this into a clinical picture, a clinical context, to see how we would use, say, levels of TSH and thyroid hormone to make a diagnosis. So just to kind of give you some simple examples, we could draw some blood, measure TSH, and measure thyroid hormone levels. Now we can measure total thyroid hormone levels, like total T3 and total T4 levels, but that would include what was bound to protein as well as what was free. So, and that could have other issues which we won't get into at this time. But if we take a look at the free is just really meaning we're looking at just the unbound T3 and T4, what's not actually bound to proteins. So let's take this first example. If I take a look and I have an increased TSH in this patient, and I have a reduced free T3, T4, logically that shouldn't make a whole lot of sense, because if the TSH levels are high, then the T3 and T4 levels should be a little bit higher, but they're low. So the low T3 and T4 is giving us hypothyroidism. And the increased TSH, well, that's a normal response to a low T3 and T4. So this is a normal response, but these levels are still low, producing hypothyroidism in this particular patient, probably giving us the clinical pictures, or a clinical picture of hypothyroidism. The defect is going to be in the thyroid gland, because the TSH level is elevated, which it normally should be, but the T3 and T4 is too low, which means the thyroid gland is probably not responding to the TSH appropriately. So therefore the person has hypothyroidism with an elevated TSH. How about this example? The person also has an increased TSH, but the T3 and T4 are elevated. Well, now the problem is, yes, the T3 and T4 are elevated, which is causing hyperthyroidism, but why is it not suppressing the TSH? So that means the problem is not in the thyroid gland, but most likely it's probably somewhere along the other axis in, say, the pituitary gland, or the hypothalamus. This last example, there's a decreased TSH and an increased T3, T4. So again, increased T3 and T4 is probably giving us our hypothyroidism, but there's a decrease in the TSH, so this elevated T3 and T4 is suppressing TSH. That's normal. It should suppress the TSH. However, with the suppressed TSH, the T3 and T4 levels should drop. However, our patient has hypothyroidism, so these levels have been retained despite the fact that it's suppressing TSH, which means that there's a problem in the thyroid gland, right? Either maybe, let's say, some sort of an adenoma that's producing this T3, T4, or Graves' disease, where there's the thyroid stimulating immunoglobulin, the TSI, which is acting independently and causing an elevation in the T3, T4, in which case you can do a blood test for that. So this is just a sort of a simple example of how we might actually use some of this information to make a diagnosis. And again, we would put this in context with the whole clinical picture. All right, so that's going to end the introduction to the endocrine system as well as the thyroid gland. But I will be lecturing on the other endocrine organs in the other videos. Okay, so take care.